Hello everyone, welcome back to Studio Students. In this video, we'll talk about Shigella. Mainly two things, we'll talk about pathogenesis and clinical symptoms. And with that, let's begin. Start with pathogenesis. First one is mode of transmission. How does it spread? It spreads from contaminated hands, or it can spread by consuming contaminated food and water. Now talking about infective dose, it can take as low as 10 to 100 bacilli to infect a particular person. Here, count with me. This is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It can take as low as 10 bacilli to infect a particular person. That's a bit scary. After ingesting the bacteria, it goes straight to the small intestine. It gets engulfed by M cells. Now you may ask, what are M cells? M cells are specialized antigen sampling cells present all over Peyer's patches in the intestine. Their main function is to transport microbes and antigens from the intestinal lumen to the underlying human cells, but Shigella exploits this mechanism to cross the intestinal barrier. After getting engulfed by dam cells, it reaches the underlying macrophages. Normally, the function of macrophages is to destroy bacteria to phagocytosis, but Shigella escapes the destruction by lysing the phagosome before lysosomal fusion can occur. It then induces macrophage apoptosis, leading to release of cytokines such as interleukin-1. This interleukin-1 recruits neutrophil, and this leads to inflammation in the intestinal mucosa. Release of interleukin-1 recruits even more neutrophils. As these neutrophils migrate across the epithelium, they disrupt the tight junctions between two epithelial cells, thus allowing some bacilli to migrate through the disrupted tight junction. At the same time, it also invades adjacent epithelial cells directly. After invading the epithelial cell, it begins multiplying within the cytoplasm. But it doesn't remain confined to a single place. It hijacks the host cell actin and causes F-actin polymerization. This forms actin tails that propels the bacterium into adjacent epithelial cells. As Shigella continues multiplying and spreading from cell to cell, extensive epithelial damage occurs. The infective cells die out due to inflammation and intracellular bacterial spread. As intestinal lining gets damaged, fluids leak into the intestinal lumen. This leads to loss of water and electrolytes, contributing to diarrhea, dehydration, and bloody mucoid stools. So let's summarize the entire pathogenesis of Shigella. After ingestion, Shigella survives the acidic environment of the stomach and reaches the intestine. It then targets the specialized M cells present over the Peyer's patches and crosses the intestinal barrier. After entering the submucosa, Shigella is engulfed by macrophages. But instead of getting destroyed, it lyses the phagosome and induces macrophage apoptosis. This leads to release of interleukin-1 and recruitment of neutrophils. As neutrophils migrate across the epithelial cells, it disrupts the tight junctions between them, allowing Shigella to invade epithelial cells. Inside epithelial cells, Shigella multiplies and spreads from cell to cell through F-actin polymerization. This extensive intracellular spread causes epithelial cell death, inflammation, and fluid loss, ultimately leading to bloody mucoidal diarrhea and dysentery. After an incubation period of around 1 to 4 days, that is Monday, Tuesday, Wednesday, and Thursday, the infected person enters the initial phase of infection. At this stage, Shigella has already invaded the intestinal mucosa, triggered macrophage apoptosis, and released cytokines such as interleukin-1. Infected persons initially develop watery diarrhea and fever. But why? This occurs because of inflammation and epithelial damage, which causes leakage of fluids and electrolytes into the intestinal lumen. And fever develops due to cytokines like interleukin-1 acting on the hypothalamus. Infected persons may also develop vomiting due to GIT irritation. Anorexia and malaise occur as part of body's systemic inflammatory response. So the symptoms directly reflect inflammatory processes initiated during the pathogenesis of Shigella infection. As the infection progresses, infected persons enter the dysentery phase. In this phase, there is a frequent passage of bloody mucopurulent stools with abdominal cramps. Now, here's the endoscopy picture which shows the blood and mucus in the intestinal mucosa. Now, let's look at the phases of complication. First one is intestinal. And here, the colon dilates because of continuous inflammation. This leads to toxic megacolon. The second one is rectal prolapse. How does it happen? Because of continuous toilet trips, it strains the rectal wall, leading to prolapse. And lastly, it is intestinal perforation. How does it happen? Because of epithelial damage and deep ulceration, it leads to formation of holes in the intestines. Coming to metabolic complication, first one is hypoglycemia. But why does blood glucose level fall? It is due to severe infection, poor oral intake, vomiting, and diarrhea. The body rapidly consumes its glucose reserves. This leads to hypoglycemia. Now, talking about hyponatremia, it's the same reason because in diarrhea, there's a huge loss of sodium, which leads to hyponatremia. Now, coming to dehydration, it's quite obvious. There's a loss of water through diarrhea and vomiting. Another severe complication is Ekiri syndrome, mainly seen in children. It is a toxic encephalopathy caused by severe Shigella infection. Infected persons may develop seizures, alter consciousness, and cerebral edema. This occurs due to severe systemic toxicity, inflammation, and metabolic disturbances caused by the infection. Patients having HLA-B27 gene develop post-infectious phase, which is only seen after as flexneri infection, which causes arthritis, ocular inflammation, and urethritis. And with this, we come to an end. I hope you understood the pathogenesis and clinical symptoms. Thank you.